The overall goal of this procedure is to provide an efficient method for generating plasmacytoid dendritic cells, or pDCs, for the study of their development.
This method can help key questions in immunology field, such as how is pDC development regulated. The main advantage of this technique is that it allows the use of a small number of progenitor cells to efficiently generate large quantities of pDCs. The implications of this technique extend toward the therapy of pDC-associated autoimmune disease such as systemic lupus erythematosus and psoriasis. Demonstrating the procedure today will be Chang, a grad student from my laboratory.
On the day before the bone marrow cell harvest, seed AC-6 stromal cell line cells at 5.9 times 10 to the 4th cells per well into individual wells of 12 well plates, and incubate the plates at 37 degrees Celsius and 5% carbon dioxide overnight. The next morning, gamma irradiate the AC-6 cells at 3000 rad and replace the culture medium with 1 milliliter of complete medium. Next, place the appropriate number of adult wild-type C57 Black 6 mice in a dissecting tray and sterilize the animals with 70% ethanol. Then move the tray to a semi-sterile hood and make incisions in the mid-abdomens of each animal, removing the skin from the distal parts, including the lower extremities. To release the